Genetic engineering is a radical technology that allows us to take bits of DNA and genetic traits from insects, bacteria, viruses, even humans, and bring those into our food crops. Why? Because if you can teach a tomato, for example, to produce enough flounder blood, that tomato becomes frost resistant and hence more profitable. It's very interesting to me that the FDA's own staff scientists have warned about unique health hazards of genetically engineered foods. And yet the FDA bureaucrats, who are political appointees, are ignoring the recommendations of their own scientists and are rushing these foods to market.
In 2012, the FDA gave its okay for a new kind of Atlantic salmon engineered to have higher levels of growth hormone using the genes of Pacific salmon and an eel-like fish called the ocean pout. It's the first GM animal to be approved for human consumption.
There's nothing substantially equivalent about genetic engineering, where you're inserting a gene just like a virus does. In fact, you even have to have virus code protein genes and a number of other things to get it to express itself. You don't have the normal controls and regulatory genetics that go along with that natural sexual process and the genetic engineering that we would normally have to keep all of those things in balance and in a controlled state.
There is no human safety testing that has ever been done on BT corn or any other GM food, so we, the consumers, are being used as guinea pigs.
What we're very good at in biology is we're very good at characterizing these individual pieces. And so this is a container filled with Lego blocks, and I have all these individual pieces. I can count them, I can ask what the distribution of colors is, and so forth. But of course, what we really want to know is how biological systems function, like this Lego engine, and parts are going to turn with respect to other parts and so forth. And of course, what's noteworthy in biology is that not only do these biological systems have interesting functions, but of course they self-assemble themselves and they repair themselves and all these other things that of course a simple object like this made of Lego blocks can't do. And so what I'll do at this point is, that was a little bit of a general introduction. What I'll do at this point is focus on three very specific problems that I think it would be very useful to actually figure out. So problem number one is relating genes to cell types. And I'll clarify what I mean by that in a moment. So how many genes do you guys have, right? We have, roughly speaking, the same number of genes as many other animals, so we're talking about 23,000. Only a small portion of our genome actually codes for proteins. The 99 or the 98.5% that doesn't code for proteins was once called junk.